BS Zoology, 3rd semester. Today we are here to discuss class BIVALVIA. As I have discussed before, at least 30,000 species have been discovered in class BIVALVIA. That is the reason it is considered as the second largest molluscan class. The animals which are found in this class are clams, oysters, mussels, and scallops. If want to know about its basic structure, then a sheet-like mantle and a shell that is consisting of two valves cover these laterally compressed animals. Many species are edible, but some form precious pearls as they are filter feeders. That is the reason they are valuable in removing bacteria from polluted water. Now come on shell of class BIVALVIA. The two convex halves of the shell are called valves. Along the dorsal margin of the shell is a proteinaceous hinge and a series of tongue and groove modifications of the shell. These modifications are called the teeth. Teeth are used to prevent the valves from twisting. The oldest part of the shell is umbu. Umbu is a swollen area near the shell's anterior margin. The shell is continuous along the dorsal margin, but the mantle in the region of hinge secrete relatively greater quantities of protein and little calcium carbonate. The result is an elastic hinge ligament. The elasticity of hinge ligament opens these valves when certain muscles relax. Adductor muscles at either end of the dorsal half of the shell close this shell. The bivalve mantle attaches to the shell around the adductor muscles and near the shell margin. Pearl formation, the unique characteristic of class BIVALVIA. If a sand grain or parasite lodges between the shell and the mantle secrete nacre around the irritant, gradually forming a pearl. Pacific oyster usually form the highest quality pearls. Respiration. Respiration have been observed in class BIVALVIA with the help of the gills. Gills form folded sheets. These are called lamellae. These are present with one end attached to the foot and the other end attached to the mantle. The mantle cavity ventral to the gills is the inhalant region, and cavity dorsal to the gills is the exhalant region. Cilia move water into the mantle cavity through an incurrent opening of the mantle. Sometimes this opening is at the end of a siphon. Now question arises, how this siphon will be formed? So this siphon is just an extension of the mantle. A bivalve buried in the substrate can extend this siphon to surface and still feed and exchange gases. Water moves from the mantle cavity into the small pores in the surface of the gills and from there into the vertical channels in the gills. These vertical channels are named as water tubes. In moving through water tubes, blood and water are in close proximity, and now gases exchange by diffusion. Water exits the bivalve through a part of the mantle cavity at the dorsal spect of the gills. This area is called the suprabranchial chamber. Now discuss the digestion of class Bivalvia. Bivalve adaptations to a sedentary filter feeding lifestyle. This type of lifestyle includes the loss of head and radula, and except for a few bivalves, the expansion of the cilia-covered gills. The gills trap food particles brought into the mental cavity. The food trapping mechanism is still uncleared, but once food particles are trapped, cilia move them to the gill's ventral margin. Cilia along the ventral margin of the gills then move food towards the mouth. Cilia covering leaf-like labial palps on either side of the mouth also sort filtered food particles. Cilia carry small particles into the mouth and move larger particles to the edges of the palps and gills. This rejected material is pseudophysis. Pseudophysis falls or is thrown onto the mental and a ciliary tract on the mental. Mental transports the pseudophysis posteriorly. Food entering the esophagus entangles in a mucoid food string, which extends to the stomach and is rotated by cilia lining the digestive tract. Consolidated mucoid mass, the crystalline style, project into the stomach from a diverticulum, and this diverticulum is known as style sac. Enzymes for carbohydrate and fat digestion are incorporated into the crystalline style. Cilia of the style sac rotate the style against a chitinized gastric shield. This abrasion and acidic condition in the stomach dislodge enzymes. The mucoid food string winds around the crystalline style as it rotates, which pulls the food string further into the stomach from the esophagus. This action and acidic pH in the stomach dislodge food particles in the food string. Further sorting separates fine particles from undigestible coarse material. The latter are sent onto the intestine. Partially digested food from the stomach now enters our digestive gland for intracellular digestion. Cilia carry undigested waste in the digestive gland back to the stomach and then to the intestine. The intestine empties through an anus through the excurrent opening, and excurrent water carries feces away. Now circulation. Circulation in case of class Bivalvia. Bivalves have an open circulatory system that bathes the organs in hemolymph. The heart has 3 chambers, 2 auricles receiving blood from the gills and a single ventricle. The ventricle is muscular and pumps hemolymph into the aorta and then to the rest of the body. Excretion. 2 nephridia are below the pericardial cavity. Their duct system connects to the coelom at one end and opens at nephridiopores in the anterior region of the suprabronchial chambers. Nervous system. The nervous system of bivalves consists of 3 pairs of interconnected ganglia. Ganglia associated with the esophagus, foot and posterior adductor muscles. The margin of the mental is the principal sense organs. It always has sensory cells and it may have sensory tentacles and photoreceptors. Reproduction. Most bivalves are dioecious. It means male and female can be observed in different animals of the same species. A few are monoecious when both male and female parts are present in the same animal. And some of these species are protandric. It's the second category. Such type of species have male parts in earlier stages of their development, but female parts can be observed in later stages of their life. Gonads are in the visceral mass, where they surround the looped intestine. Ducts of these gonads open directly to the mental cavity or by the nephritopore to the mental cavity. Most bivalves exhibit external fertilization. Gametes exit through suprabranchial chamber of the mental cavity and the exhalant opening. Development proceeds through trochophore and veliger stages. When the veliger settles to the substrate, it assumes the adult form. Most freshwater bivalves brood their young. Fertilization occurs in the mental cavity by sperm. These sperms brought in with inhalant water. Some brood their young in maternal gills through the reduced trochophore and veliger stages.